All right, it started.
Well, good morning, everyone. I'd like to welcome everyone to Lucky Session 13 of the 2022 IEP Annual Workshop. We've got a fun set of panelists today. I'm looking forward to all of their talks. And I wanted to do the usual starting session reminders, which is if you see something you like, please use the hashtag on Twitter or Instagram or whatever, 2022 IEP Workshop. If you have questions, it's the same drill. Please either type them into the Q&A box that you'll find in your Zoom menu, or raise your hand and we'll address all the questions at the end of each individual talk. I just want to point out, I think, not 100% sure, but I think this is the session with the most taxonomic diversity. So pretty excited to see what all the panelists are going to tell us today. And starting us out with a topic of very high interest to a lot of people is invasive vegetation in the Delta. And we've got Drs. Anastasios Mazis and Bailey Morrison, both at UC Merced, doctoral scholars in Aaron Hester's lab. And they're going to be telling us today about water primrose.
Sorry, just getting everything switched
Hello,
over
everyone.
to notes.
My name is Dr. Bailey Morrison, and I'm co-presenting today with Dr. Tassos Mazis. We are both postdoctoral researchers at the University of California, Merced, working with Dr. Aaron Hester in the Earth Observations and Remote Sensing Lab. Today, we're going to present research we are conducting with our partners from the U.S. Geologic Survey, the California Department of Fish and Wildlife, and the University of Santa Cruz that is focused on gaining a deeper understanding of how water primrose is invading emergent marshes in the Sacramento-San Joaquin Delta. So to get started with some background, water primrose is a very successful invasive amphibious aquatic macrophyte that was introduced into the Delta in the early 1900s. In some previous research from one of our co-PIs, Dr. Shruti Khanna, who's with the California Department of Fish and Wildlife, she found from 2004 to 2016, water primrose cover has increased fourfold over a 12-year period. And she did this analysis using archival hyperspectral airborne remote sensing data. And just to point out that since 2016, when this study's time range concluded, water primrose expansion has only gotten worse and been blowing up even more. And so the figure that we're showing below is from Dr. Khanna's manuscript over Liberty Island from this 2004 to 2016 period. There's a clear increase in water primrose, which is shown in yellow, with some reductions in water hyacinth, which is another invasive species, submerged aquatic vegetation, and open water habitat during that time period. So additionally, in the same study, Dr. Khanna performed a change detection analysis and found that after water primrose establishes itself in open water habitat, it just starts to continue to expand rapidly outward from there, but not actually going into these emergent marshes. However, once it's been in this open water habitat and completely filled it and exhausted the habitat, it, instead of normal species where when it runs out of its optimal habitat, they kind of get restricted to that distribution, the water primrose will actually switch the direction of spread and encroach into emergent marshes, even though these habitats are less suitable, and it's starting to replace native emergent vegetation like tule and cattail. So this has some pretty important ecological and restoration management implications. So ecologically, water primrose invasion into emergent marshes can lead to overcrowding and outcompeting both native and non-native species. So hyacinth in the past was a dominant invasive species, but water primrose now appears to be the more dominant species and much more aggressive and actually pushing hyacinth out in some areas. Water primrose invasion can also lead to altered functional relationships with emergent species, cascading community effects, and water primrose invasion may actually reverse the rate of succession and knock marshes out of their successional equilibrium. In terms of restoration management and overall delta management, water primrose encroachment into emergent marshes may reverse successful restoration projects that have already occurred in delta habitat, and it also could threaten future restoration projects. If marshes also become more unhealthy due to water primrose invasion, more regulations on water usage for water coming out of the delta could end up being imposed. And then lastly, water primrose is largely treated and managed using herbicides in their open water habitat they've traditionally been in. So if they keep spreading into these emergent marshes, we don't actually know whether or not we can spray in the marshes to kill water primrose without actually harming the native species that we're trying to protect. So given Dr. Konda's previous research we just went through, and we know that it's dramatically increasing in cover in the delta, and now it's starting to invade into these emergent marshes, or we don't know. Okay, so we know that they're increasing in cover, and we know they're now invading marshes. But what we don't know is the mechanisms and the drivers of water primrose invasion on marsh loss and vulnerability given all this. And so given all this information, we're able to receive California Prop 1 funding to try and answer and fill in these knowledge gaps. And our project's going to be focused on first identifying functional traits of water primrose that are linked to marsh loss. And secondly, we're going to determine the effects of biodiversity on water primrose invasion success and the occurrence of marsh loss throughout the whole delta. We're planning on accomplishing these two tasks by measuring and analyzing plant traits, allelopathic chemicals that are produced by water primrose. So allelopathy is an organism or a plant's ability to create chemicals that can inhibit another organism's success and survival. Water primrose knows to do that, so we're including this in our analyses. We're also going to be collecting environmental DNA, or eDNA as it's known, to measure and model biodiversity and species presence at our field sites and throughout the delta. And we're using eDNA because it contains DNA from things like fecal matter, plant roots, bacteria, and funguses, and much more that are able to fully or have a better representation of biodiversity in these marshes versus just doing standard plant vegetation diversity metrics. And along with the eDNA, we're also going to be genetically identifying water primrose species. So it's known that there's 3 different water primrose species in the delta right now. There could be up to 6 different ones, though, given expert information from Dr. Brenda Newell. And so we need to know if any of the differences we're seeing are due to phenotypic or genotypic differences in the same water primrose species, or if we're dealing with completely different species altogether. And then lastly, we're also going to be using archival hyperspectral airborne imagery and drone surveys to assist with all these other analyses and model and tie everything together. So with all of these different data types, we're then going to quantify the amount and spatial trajectory of marsh loss from 2004 up till 2020. And then lastly, we're going to synthesize all the data together to identify and map which marshes are most vulnerable to water primrose, as well as give a ecological value to each of these marshes, and they're going to be our final public data sets that people will be able to use to help with management and restoration projects. So to date, we've already completed 2 of our field campaigns. We have 1 more planned in a few weeks in mid-April of this year. And so now Dr. Mazis is going to discuss and present some of the early results that we have from these first 2 campaigns and the analysis that we were able to complete. So here you go.
Thank you, Dr. Morrison. And as Dr. Morrison just said, we have a bunch of data. So in this presentation, we will focus on some of the morphological and physiological traits that we have collected so far. The morphological traits were measured on the field with hands-on equipment, and the physiological traits were estimated using optical-based measurements and with calculated vegetation indices. And also, we have the different study factors, and as we see here, we have treatment, so we did measurements in patch and interface environment. We have different season, we took measurements in July and November, and different salinity levels, we took measurements in freshwater sites and saltwater sites. So after we measured the variables, we wanted to see which ones show significant difference between the different levels of the factors. As we can see here, the rows show the measured traits that describe the morphology of the plant. And we also have some physiological parameters like light use efficiency, lignin, and chlorophyll that they were derived with spectroscopy methods. In columns, we have the different factors of the study. So from left to right, we have treatment, interface and patch, season, summer and fall, and salinity level, freshwater and salt water. As we can see, plant height and internodal distance showed significant differences between interface and patch. When we talk about internodal distance, it's basically the distance between different nodes on the plant stem. And the plant height and internodal distance shows significant difference between summer and fall as well, and between different salinity levels. Significant differences were also identified for light use efficiency, lignin, and chlorophyll, with them showing significant difference for all levels of all factors as well. Leaf water difference didn't show any significant differences, and this stems from the abundance of water in the system. So the plants, in reality, they aren't water limited. But knowing that the variables show significant difference doesn't tell us much. We want to actually know which level of the factors favor water primrose growth more. So if we take a more detailed look on the different factors of the study, we can see here the effect of season on the morphology and physiology of the water primrose. And we see that the height, the nodal distance, and chlorophyll, they were higher in the summer compared to fall measurements. Also, light use efficiency was higher in the fall. That makes sense physiologically, since summer is the active growing season, so we have bigger plants. And as the day becomes shorter in the fall, the light use efficiency is increasing. Similar results we have when we look at the effect of salinity on the plants. The morphological traits, which are height, the nodal distance, and leaf area, as well as the chlorophyll, are higher in the freshwater sites, where the light use efficiency is increased in the saltwater sites. Last, when we look at the effect of treatment on the plants, we see that plants that they are growing on water, so they are patch for this study, they grow more in a horizontal manner, and they invest more in leaf area and greener leaves. And also we see higher amounts of lignin. Because they grow without shading, the light use efficiency is typically lower compared to interface plants. There, in the interface, water primrose has to compete with native vegetation, mainly tule and cattail, which are generally taller plants. So as a response, we see water primrose getting taller with larger internodal distance, investing more in a vertical growth in order to take advantage of the available light. For that reason, and because shorter plants are out-competed and they're getting shaded, we see higher light use efficiency, since the plants have to utilize all available water. Light. What is the take-home message of our results? The environment water primrose grows in affects the condition of the plant, both the morphology and the physiology. And as a result, interface leads usually to taller plants. The traits also differ between seasons, meaning that we have an active, inactive growing season, fact that will help us time our management practices better. And last, freshwater seems to favor water primrose growth, a factor that it will be more and more important as we see a gradual change in salinity levels in areas in the delta. So who should keep an eye on our findings? First of all, the stakeholders and the policymakers can utilize our results for more informed decision making that will lead to a more efficient and more effective management of the delta. But our findings will also be useful to the scientific community, since they will help us understand more about the physiology and the morphology of water primrose, as well as assess the growth strategies of the species and how this favors further expansion. Thank you for attending our presentation. Here we have the email of Dr. Morrison, Dr. Mazis, and Eeyore's lab. So please feel free to reach for any further questions.
Thank you.
All right, thank you, Anastasios and Bailey. Really interesting talk about something that's all over our marshes these days. If anyone had questions, this is a good time to type them into the question and answer box, or to raise your hand and we can call on you. And if not, I get to ask all the questions. So the first question that I had is probably the one that nobody working in invasive vegetation wants to answer, which is, if we can't do open water herbicide on water primrose, what do we do to manage it?
I don't know if people don't want to answer that or if we have no clue what to do in that case. I know that the work that we've been doing, and I also worked as an undergrad in Dr. Brenda Gruel's lab. There's been a lot of work looking at like nitrogen and carbon and resource allocation and effects of light on water primrose to try and see if we can find any weaknesses that we can use to our advantage to try and maintain it without herbicides. But I'm not sure if there's been any kind of long-term broad ways to try and manage it without herbicides, or something else that's going to be effective at actually managing it at this point. So we still have a lot of stuff to try and dig through and see if we find anything, but yeah, that's going to be a big issue if the herbicides end up not working as well.
We've got, thanks, Bailey. We've got one question in the chat from Gina Darren asking, what's the status of biocontrol agents with water primrose?
That I'm not sure about. We're not specifically looking really at how the herbicide treatments are impacting water primrose. Our studies way more focused on just trying to understand how it's able to invade into these terrestrial areas. We are interested, I guess one type of biocontrol is we're doing this eDNA, the environmental DNA analyses. We're trying to understand if there's going to be like, if biodiversity resilience is basically a factor. So it looks like there's some marshes that aren't really being invaded while there's others that are. And we're hoping that the biodiversity resilience may be something that's allowing these marshes to basically resist being invaded. And if that ends up being the case, then restoration and conservation efforts that would help boost the diversity of these marshes would definitely have the potential to help control water primrose spread.
It is the hope that bolstering the natives does help control the invasives. And I know that there's some research happening on that with Gina Darren, at least. I think we've got time for one last really quick question, mostly for Anastasios before we go on, unless anyone else has a question they want to ask. I noticed that there was more lignin in open waters. Can you explain really quickly why?
Yes, so keep in mind that these are the preliminary results. So the next field campaign might give us some results that it will make us rethink some of the existing results. Our focus on the first manuscript that it will come out from this study focus more on the allocation of resources as for leaf area and lignin. So our guess is that maybe in the open area, the plants that don't need to allocate resources into getting taller and thinner. So maybe all the lignin, it's allocated more in the leaves. So since we took, since most of the measurements they are focused on the leaves, maybe we saw some more lignin in them, in the open water samples. Our goal, and we are thinking about it, but the logistics is a little challenging with this kind of studies, is to take some measurements maybe on the whole plant. So this might change our perception about where it allocates lignin more.
All right, sounds interesting. Good to know that you guys haven't run out of research questions to pursue on water primrose.
The irony is that the more we study about that, the more questions we have. So we hope that we will get to answer most of them at least.
That is what seems to keep happening with invasives generally. Well, thank you guys. That was a really interesting presentation. If I can change into an entire kingdom to the next talk, I'd like to welcome Caroline Newell, who's a junior specialist at UC Davis, working in the Moyle Duran lab. She's done fish research in Suisun in the past, but today is going to be talking to us about the Black Sea jellyfish and what it's doing up in Suisun Marsh. And Caroline, I think you're doing this talk live, which means, Stephanie, you might need to give her permission to share and activate her camera.
She should be able to.
Okay, I'm sharing now, and then it says I'm not able to share my camera.
Okay. We do see your presentation?
Yeah.
Okay, great.
Yeah. So we can just roll like this.
Give me one quick sec.
Oh, okay.
In the interest of time, I think we should just go ahead and go. Sorry, I'm having a little trouble.
Okay. Yeah, no worries. So hello, everyone. My name is Caroline Newell. As stated, I am a junior specialist in the Peter Moyle John Duran Lab at UC Davis in the Center for Watershed Sciences. And I will be talking to you today about Black Sea jellyfish, their trends and life history in Suisun Marsh. So in my role as a biologist, I sometimes get to engage in science communication. Back in 2020, I was given the opportunity to write a science communication piece for the Frontiers for Young Minds journal, and I chose to write about work done by Alpa Windsor, Mariah Meek, and Peter Moyle on the trophic ecology of Black Sea jellyfish in Suisun Marsh. So I put this out there, and then organizers for the session read it and asked if I could talk about the work done about Black Sea jellyfish in the San Francisco Estuary, Suisun Marsh area, and the trends that we've seen over time. And so that is to say, I'll be using this talk to introduce you all to Black Sea jellyfish and talk about their presence in a special part of the San Francisco Estuary. That special spot is Suisun Marsh. You can see over here, I have a kind of general outline of the San Francisco Estuary. And over here, we have the Pacific Ocean on the west side, bringing in salty water. On the east side, we've got the Sacramento-San Joaquin Delta, bringing in fresh water, and Suisun Marsh lies right in the middle of that. So it's brackish, and it varies in salinity depending on the time of year and the amount of fresh water or salt water it's got coming into it. So why did I choose to write about Black Sea jellyfish? Well, the reason I was interested in jellyfish is because I grew up in Fairfield, located right above Suisun Marsh, and I never knew we had jellyfish there. Here's little old me. In 2004, I was 8 years old on a field trip to Suisun Marsh. And so I grew up there, never really thought of the marsh as being like anything oceanic, I guess. And then flash forward to 2018, and I got to start working for the Moyle Duran Lab at UC Davis. And our lab samples the marsh every month for the Suisun Marsh Fish Survey. The survey has been going since the late 1970s and documents fish assemblages found in Suisun Marsh over time. The first time I got to go out and sample the marsh, I was surprised by a lot of things, but totally shocked to find jellyfish there. Here's a picture of me holding a jellyfish in 2018 on the survey. And I was, totally delighted, but totally shocked. I had always thought of jellyfish as being like this oceanic thing and had only ever seen them in tanks and aquariums and on like nature documentaries about the ocean. And so I was kind of wondering, like, how did we end up with jellyfish way up here, and where did they come from, and what do they mean for our system? And so I dug into a lot of literature and got to learn everything I could about these jellyfish. And I find that jellyfish are inherently interesting, but there's a lot of research on them because they are non-native and a potentially invasive species. So the San Francisco Estuary has many non-native species, and some of them tend to be damaging or invasive. And we have 3 species of hydrozoans in the estuary. The reason that I am talking about Mazis marginata in particular is because that's the one we have the most data on for the Suisun Marsh fish study. So it's the one I can talk about the best. There are 2 other species, so Blackfordia virginica and Mauritia leonsi. These 2 other species aren't caught very well by our sampling methods. So Black 40 virginica need more salinity, and so they are not really in the marsh, which doesn't have enough salinity for them. And Mauritius Leon Sea are too small for our nets to catch. So they may also be impacting the system, and they may be following similar trends as Mauritius marginata, but I can't really speak to that in this presentation. But yeah, so that's the reason that we're focusing on Black Sea jellyfish, non-native, potentially invasive, and I have the data for them. General outline for the rest of this talk. I'm going to introduce you to the basic biology of Black Sea jellyfish, and then talk about Black Sea jellyfish in Suisun Marsh. Their long-term trends, water quality relationships we found, trophic ecology, and potential management for the species. So what are Black Sea jellyfish? Well, they are a jellyfish with many life stages. They have their egg/sperm stage. They can be polyps, planuloid, polyp buds, resting pedal cysts, and medusae. You don't have to know what all those are specifically, just know that 4 of those life stages are capable of reproducing and being transported to new places to reproduce. So polyps, planuloid, polyp buds, resting pedal cysts, and medusae. I bolded the planuloid polyp buds because those are, that's like something special, kind of special about Black Sea jellyfish. They're one of a small number of species where the polyps can also reproduce by budding small planuloid bits of tissue known as the polyp buds. They were first, we believe that they were introduced in the 1950s and that by ballast water from the Black Sea, hence the name Black Sea jellyfish. And there's potential, it's possible that there have been multiple rounds of introductions. To give you an idea about their size, their max bell diameter is about 50 millimeters, kind of like the size of a golf ball. They eat a wide variety of planktonic organisms and have been documented to eat larval fish as well. They have a unique and effective feeding style. Here on the left is a diagram I made based on Winsor and Mills and Sommer's papers describing their behavior. They'll lie in a feeding spot on the substrate, and when they decide to switch spots, they will flip over, actively swim to the top of the water column, flip over again, and then drift feed down with their tentacles outstretched, feeding on pelagic organisms until they reach another spot because the current pushes them, and they will feed there. And so planktonic organisms are captured during their descent, and once they contact the substrate, they'll remain ex-umbrella down while twitching the ends of their tentacles, kind of like a luring behavior. Medusae can have up to 600 densely packed tentacles around the bell, and they have specialized nematocysts that are effective at penetrating hard surfaces, such as chitinous exoskeletons and scuds. So now you know a little bit more about these guys. Let's look at the data we have on them. Here's some data from the Suisun Marsh fish study. The y-axis is water year, and the x-axis is water year. Y-axis is catch per minute trawl for all trawls done in the survey. And we see that the time series it begins in the late 1970s. Black Sea jellyfish were likely already around by then. And so we don't know what they were doing before that. But since 1980, there were kind of like little blips here and there where they showed up, but they weren't really not continuously, but year to year present since around the year 2000. Since then, there has been an overall increasing trend of Black Sea jellyfish in Suisun Marsh, with our max number of individuals caught. In 2019, we had 21,154 individuals, which is pretty crazy. And keep in mind, these are only medusae catches. We don't have data on what the polyps are up to. So what's driving these trends? You may be looking at this and think why are there huge booms one year and not a really huge boom the next year? Well, when we look at seasonality, we kind of get an idea of what's going on. So this is the same graph as before, but broken up by season. And the winter seasons are January to March, spring, April to June, summer is July to September, and fall is October to December. We see a lot of the yellow and green here, indicating favorable conditions for Black Sea jellyfish are happening in the summer and in the fall. As we have longer summer and fall periods, we are likely to get more jellyfish in the system because there's longer periods favorable to them. The length of time and abundance of jellyfish in Suisun Marsh has to do with two critical abiotic conditions, temperature and salinity. So the reason we see them more in the summer and fall is because that's when it's warm and salty. Here are 3 plots on this slide, one showing average salinity parts per thousand by month in the marsh over the years for Suisun marsh fish study. Here's water temperature by degrees Celsius average over the Suisun marsh fish study by month as well. And here is the catch per minute trawl broken up by month for all the years that we've done sampling. So we see that we start to catch them in June, more in July, then August, we hit a peak, September it peaks, and then they start to decline October, November, December. And if we look at the temperature and salinity, graphs, we see in June, that's when salinity, when we start catching them is when salinity starts to pick up and the temperature is really high. Then in August and September, our peaks are happening when salinity is high, temperatures are high, and they start to kind of get beaten down as the temperature cools and the salinity drops as well. So if it's sunny and dry and the temperature is high, we get more jellyfish. Then when it gets rainy and cool, the amount of jellyfish goes down. The Schroeder 2011 stated that their 3 to 10 PPT is ideal for them. The extreme ranges that we found them is between 0.4 and 15 parts per thousand. And we most commonly catch them between 3 and 10, 2.7 to 10.5. The max range of water temperature we found for them was between 8.3 and 28 degrees Celsius, with the most caught between 14 and 23.4 degrees Celsius. What impacts are these jellies having on the system when they're huge with their huge summer and fall blooms? One theory is that they're competing for food resources with pelagic fish species. The annual increase of Black Sea jellyfish has occurred during the same time period as the crash of pod fishes, which I've outlined in purple. One proposed reason for the crash is that of pelagic organisms or fish is that they have not had enough food, and competition with invasives is listed as a likely cause. Summer and autumn is when pelagic organism decline fishes most need plankton, and that is also when jellyfish are the most abundant in eating those planktonic foods. So there's fear of direct predation as well. Are the jellyfish eating these fish, and are they also competing? These questions drove Alpa Windsor, Mariah Meek, and Peter Moyle to carry out a diet study on the jellies in Suisun Marsh. In 2007-2008, they analyzed the gut contents of 200 individuals and found that jellyfish ate 13 different taxa of prey over the bloom period. They were found to mostly eat calanoid copepods and barnacle nauplii, which is supported by a diet study done by Mills and Sommer in 1995. As shown to the right, the calanoid copepods they were mostly eating were Acartiella sinensis and Pseudodioptimus for Betsy. Jellyfish also ate cyclopoid copepods, chiropid amphipods, egg sacs, mysids, crabzoe, gams, and other small things. A couple of fish larvae were also found in their guts. Schroeder in 2011 looked to see if they might be impacting the abundance of planktonic prey and found that they are often sufficiently abundant to impact copepod abundance, particularly in interior sloughs where prey densities are moderate to low and medusa densities are high. In downstream areas in Suisun Marsh near Suisun Bay, where the pelagic organism decline has been the most severe, there was only a moderate to low levels of predatory effect. This all suggests that predation by medusa is unlikely the sole explanation for pelagic organism decline. So overall, Black Sea jellyfish had little diet overlap with pelagic organism decline fishes as well. So they never ate the pod species. The fish larvae that they had in them were Shimopheri gobies. And that has partly to do with the timing of when these things are happening. So the larval fish stages for pelagic organism decline species aren't happening when the jellyfish are abundant. However, as climate change alters conditions, Black Sea jellyfish favorable conditions are likely to be elongated, and there might start to be overlap with other fish larvae. So that means that the amount of larval fish in jelly diets might change if we see that happen. There was, when they looked at stomach fullness of striped bass and threadfin shad, two pelagic organism decline species, they found that the gut fullness was not changed over the pre-bloom, bloom, and post-bloom periods. There's also no trend found for threadfin shad. So these results do not lend support to the idea of competition for prey between fish and medusae during the bloom. However, Mauritia species, Mauritia leonsi, had some overlap with threadfin shad. How do we manage the species if we start to see them having more impacts? Running the salinity gates during the summer might work. If we beat back the salinity, then we might be able to push these guys out. So in summary, there's overall increasing numbers of Black Sea jellyfish in the estuary, and there is potential for impacting food availability for fish generally. If climate change conditions continue to increase the Black Sea jellyfish timing, the overlap with other larval fish would be very bad. And to manage this species, running the gates during the summer would be useful. More research is needed on what the polyps are doing and seeing if these results from 10 years ago still hold, because since those studies were done, we've had more jellyfish in the system. Thanks for watching this slide, and thanks to our funders at DWR and UC Davis, and thanks to my lab mates for help with this presentation.
Wonderful. Thank you, Caroline. Good to see someone shining a light on a species or a group of species that maybe don't get quite enough attention compared to the impact that they might be having. If anyone has any questions, I know I do, please ask them in the question and answer box or raise your hand. Oh, I think we've got one over there. Francis Wilkerson has asked, how does food availability vary seasonally? Is there more in summer, like the abundance of jellyfish?
I don't know the answer to that question. I think based upon my knowledge, it's possible that we are having more. So a lot of food gets put into the system when the ponds, the managed wetlands, release their waters. And I believe that happens in the summer, but yeah, I can follow up with you after asking people who know more about it and say more about like if the food is most abundant when the jellyfish are. So the jellyfish are responding to like planktonic or conditions that are typically good for planktonic food development. And that's when they are budding. But yeah, I don't know if that's actually what we're seeing in Suisun Marsh.
I see Arthur Burrows has his hand up. Arthur, do you have a question?
Yeah, quick question. You mentioned that operating the salinity control gate might have an impact on the jellyfish abundances. Is either you or anyone over at your lab looking at the data you have and comparing it to existing control gate actions and seeing if there's been any impact?
Nope. we are not super focused on jellyfish at this time in terms of our like work, and we haven't looked at the salinity control. I know that there's been work done looking at salinity control gate stuff from like John Durand and like, but I haven't actually familiarized myself with that information, and I don't know if there's been any like specific look at impacts on jellyfish and like seeing if that has an impact on their abundances.
Sounds like a great master's thesis opportunity.
Yeah. Totally.
One last quick question before we move on, very quick. Wim Kimmerer asks, have you looked at the prey compared with the open waters of Suisun Bay where jellies are uncommon?
Yeah, so yeah, it does seem that the jellyfish are like less common in those bigger waters, but it'd be interesting to look at if it is because there's less food available for them, and because we are finding them a lot, like we find them in all the sloughs, but we do see a lot of them in the dead end sloughs, like in the smaller ones, and so it's possible that they are they are responding to like higher concentrations of food. But it also is possible that they just like they don't have the correct type of substrate for their polyps and things of that nature. So yeah, that's a good question. But again, I don't know that there's been any work done on that. Sorry for not knowing the answers to these questions, but they're all interesting.
That's why we're still doing research.
Yeah.
I will say that there's a lot of interest in in jellyfish questions in the Q&A box. If people are curious, Caroline, I'm going to volunteer people to email you and ask the questions directly.
Yeah, I'll put my email in the chat.
Thanks very much. Moving on, we're we're gonna hear next from Kathy Hieb, who's telling the story of not just one, not just two, but a suite of invasive arthropods.
Thank you, Betsy. This morning I will share several stories of introduced crustaceans to San Francisco Estuary. I'm including crabs and shrimp that were introduced in the past 3 to 4 decades since the late 1980s. I'm going to discuss the when and the how, population trends, current status, and impacts, potential and realized. Included are the Chinese mitten crab, Erochir sinensis, the European green crab, Carcinus manus, the Siberian prawn, Palaemon modestus, many of you would know this as Exopalaemon modestus, the Mississippi grass shrimp, Palaemontes caudicensis, and the Asian mud shrimp, Eupogibia minor. Note that the mitten crab and green crab are on the IUCN's list of top 100 worst invasive species in the world. The Chinese mitten crab was first detected in the San Francisco Estuary in 1992 and was likely introduced several years earlier. It was probably a deliberate introduction from Asia, where it is prized as a food. Crabs are kept live until they're cooked. And this is a picture of a mitten crab vending machine in China where you can buy individual live mitten crabs. The mitten crab has been on DFW's list of prohibited species since 1986. You cannot possess or transport live mitten crabs in California. This is a picture of seized mitten crabs from airport in New York. And as recently as fall of 2019, U.S. Fish and Wildlife Service conducted an operation called Hidden Mitten and confiscated approximately 15,000 adult mitten crabs. The Chinese mitten crab population in the estuary boomed from 1997 to 2002, followed by a bust. In 1998, an estimated 750,000 adult Chinese mitten crabs were collected at the Tracy Fish facility in the South Delta during their downstream migration. Since 2006, numbers have been very low, with an estimated 12 mitten crabs from the Tracy Fish facility. This is expanded from 3 or 4. 3 in 2010, expanded from 1, and 4 in 2019, also expanded from 1 mitten crab collected. Bait stealing is also reported in these years, including fall of 2019 when bait stealing was reported from Bethany Reservoir, which is downstream of the fish facilities in the South Delta. Potential impacts of the mitten crab include burrowing in levees, clogging diversions, especially during their downstream migration when thousands of crabs can be migrating downstream, predation on salmon and steelhead eggs, and finally, the mitten crab is an intermediate host for the Asian lung fluke, with mammals, including humans, as the final host. The green crab, Carcinus manus, or the raving mad crab, was first detected in the San Francisco Estuary in 1989. It was possibly introduced with live bait from the East Coast. We know that our crabs, genetically, are from the East Coast. Green crabs have been on the East Coast for many decades, where they were introduced from Europe, where the crab is native. Note that there's also other introduced populations in the world. So relatively widely distributed species. The green crab rapidly spread north along the west coast of the USA in the 1990s to Washington and beyond, and now it is at Queen Charlotte City, BC. There is no routine monitoring in San Francisco Estuary for the green crab, but we know it is still present. We occasionally collect green crabs in otter trawl in San Francisco Bay. We know it is very common in estuaries to the north and was subject to an eradication program in Seadrift Lagoon in Stinson Beach starting in 2009. They used traps to collect hundreds, if not thousands, of green crabs. In 2009, the population was approximately 120,000. It was reduced to under 10,000 in 2013, but in 2014 they had a very large recruitment event which resulted in approximately 300,000 crabs. This is about a 3 times increase over the population that it started with in 2009. They hypothesized that there were few adult green crabs in 2014 to cannibalize the juveniles. This resulted in what was termed overcompensatory recruitment. And lesson learned about attempting to eradicate species versus control of a species. Potential impacts of the green crab include predation on shellfish, specifically commercial shellfish beds, and preying on the same intertidal clams that shorebirds depend on. In addition, there's concern over competition with native crustaceans and damage to eelgrass beds. These impacts have been reported from enclosure studies, but have not necessarily been extended to populations outside of enclosures. The Siberian prawn was first detected in San Francisco Estuary in 2000. It was introduced to the Columbia River in the 1990s, possibly with ballast water. Here it is common in tidal brackish and freshwater areas and non-tidal freshwater upstream of the delta. In the U.S. Fish and Wildlife Service Beach Saint Survey, it is most common at Knights Landing, Liberty Island, and Rio Vista on the Sacramento, and Dos Rios on the San Joaquin upstream of Stockton. It can complete its entire life cycle in fresh water and reaches a maximum size of approximately 3 inches. The Siberian prawn is now the most common shrimp in some areas of the estuary. In Suisun Marsh, it is now the most common species. Since its introduction in 2000, on average, Crangon franciscorum numbers have decreased about 50%. The numbers of the introduced Palaemon macrodactylus about tenfold, while overall annual shrimp CPUE increased. Potential impacts of the Siberian prawn include competition with the native Crangon franciscorum and the introduced Palaemon macrodactylus in brackish water. Also competition with the native Syncharis pacifica in freshwater. Syncharis is found in 17 stream segments in Napa, Marin, and Sonoma counties, including tributaries to San Pablo Bay. However, to date, there's been no overlap in the distribution of the Siberian prawn and Syncharis in the watershed. There's also concern that the Siberian prawn may compete with fishes for prey, primarily mysids, and this is because its cousin, Palaemon macrodactylus, was shown to prey largely on mysids in diet studies that were conducted in San Francisco Estuary. The Mississippi grass shrimp, Palaemontes caudacensis, was first detected in Cosumnes River Preserve in 1995. This was likely an aquarium release, as it's sold live in the aquarium trade. It's an established species, but with only occasional collections in the delta. In the U.S. Fish and Wildlife Service, beach seine has been most common at Terminus, Delta Cross Channel, and the Knights Landing stations in the central and north delta. It completes its life cycle entirely in freshwater, and this is a small species, generally no more than 1.5 to 2 inches in length. And this is a female shrimp with eggs, so this is a mature Palaemontes. I believe that the impacts of this species are minimal due to its small size and low population numbers here. The Asian mud shrimp was first collected in San Francisco Estuary in 2009. However, these first specimens were incorrectly identified as the native blue mud shrimp. Several specimens and several years later, the introduced Asian mud shrimp was correctly identified. It's possible that this species was introduced as larvae with ballast water. It's also possible that adult live Asian mud shrimp were introduced to San Francisco Estuary. It is sold live as a food in Asia. However, it's highly unlikely that live mud shrimp would have survived and reproduced in San Francisco Estuary, in part because other mud shrimp do not survive when outside of their burrows. The Asian mud shrimp is now found from Elkhorn Slough to Bodega Bay and is common in San Francisco Estuary. This in 2022, a group from UC Davis collected over 150 adult Asian mud shrimp from South San Francisco Bay in their toes. We believe that these shrimp were displaced from their burrows by low salinities that accompanied the frequent storms in December of 2021. The Asian mud shrimp is distributed in the intertidal and subtidal and constructs burrows that are up to 2 meters deep. The top of the burrow is U-shaped with a single burrow descending beneath the U. This is an ecosystem engineer, as are all mud shrimp. The burrows expand sediment surface area. Mud shrimp circulate oxygenated water into the subsurface sediments. This accelerates sediment remineralization, and also the burrows support a large number of commensal species, and some of them are obligate. The Asian mud shrimp is host to the parasitic isopod Orthiona graffinus. This is a parasitic castrator found in the gill cavity of the host. This bump is the isopod. It's under the carapace, and this is the native blue mud shrimp. This isopod has such an energetic demand that adult mud shrimp cannot reproduce, and there's recruitment failure. Orthione was first found in Willapa Bay, Washington in 1988, but upon examination of Asian materials was determined to be native to Asia. Orthione here is found from Baja California to Sitka, Alaska, and has decimated many populations of the native blue mud shrimp, Eupogebia pugentensis. This is because the native blue mud shrimp did not evolve with this parasite. Here, a high percentage of the native mud shrimp are found to be parasitized with Orthione, where the distribution of the two species overlap. And it's as many as 19 out of 20 of the blue mud shrimp collected are parasitized with Orthione. The Central California blue mud shrimp populations that overlap with Orthione and the Asian mud shrimp are now ecologically extinct. This is from Elkhorn Slough to Bodega Bay. Other populations that overlap with Orthione are either collapsed or reduced. And Orthione now extends to Sitka, Alaska, beyond the top of this map. The native blue mud shrimp was last collected in San Francisco Estuary in 2018, and the blue mud shrimp south of Humboldt Bay is genetically distinct from the blue mud shrimp populations north of Humboldt Bay. The Asian mud shrimp has contributed to the collapse of the native blue mud shrimp populations, especially in Central California. This is because the Asian mud shrimp is host to the parasitic isopod Orthione. However, is the Asian mud shrimp a replacement for the native blue mud shrimp? We know that in its native range, the Asian mud shrimp has dense burrow galleries with over 300 per square meter reported. What we don't know is that if the native commensal species, some of them obligate, will occupy Asian mud shrimp burrows. This includes shrimp, crabs, bivalves, and polychaetes. In summary, there have been significant introductions of crabs and shrimp to San Francisco Estuary in the past several decades, specifically since the late 1980s. The outcomes of these introductions have been varied. 3 of the 5 species discussed are now common. This includes the green crab, the Siberian prawn, and the Asian mud shrimp, while one species, the Chinese mitten crab, had a boom and bust pattern. Note that our fish species only occasionally collect some of these crustaceans. This is because their distribution is intertidal and shallow subtidal, areas that are not at all sampled with our towed nets. These species are associated with structure and vegetation. This is especially true for the shrimp. And some of these species occupy burrows or buried in substrate, which are also habitats that are not well sampled by towed nets. In spite of these sampling challenges, we have a very basic understanding of when and where these species were introduced, their population trends, and current status. This is thanks to sampling and data from various sources, not all California Department of Fish and Wildlife, and the Interagency Ecological Program. I'd like to thank the California Department of Fish and Wildlife, especially the field and lab staff, the Interagency Ecological Program for support and funding, TJ Arrear and Ted Grossholtz from UC Davis, John Chapman from Oregon State University, and Jonathan Spiegel with U.S. Fish and Wildlife Service in Lodi. Thank you.
Thank you so much, Kathy. That's an incredibly wide-ranging talk over a bunch of agencies, a bunch of species, and a bunch of different habitats. And I don't envy anyone working on a species you have to dig through 2 meters of mud to even count. If anyone has any questions, please do ask them in the Q&A box or raise your hand and ask them live. I know for myself, I was one of the folks hauling out traps from Seadrift Lagoon, and it's a little dispiriting to realize quite how much they didn't get eliminated. I guess as an overarching question, how much do you think some of the other species are kind of replacing ecologically the things that they are displacing?
Well, I think that that question is very relevant to this introduced Asian mud crab, or mud shrimp, I'm sorry, crab shrimp. And that is because it is common in intertidal and shallow subtidal here, very similar to the native blue mud shrimp. But, you know, the question remains, I think, and it's not well studied at all, whether or not our native commensals will occupy those burrows. So the mud shrimp burrows, when they're dense, have been described as, by John Chapman, as an inverse coral reef system. They extend so far down, there's so many of them into the substrate, and they're so significant as an ecological engineer that you know, it's certainly for us that understand what's happened, one of the more disastrous stories of unintended consequences. If even that, we don't even know if the Asian mud shrimp was a deliberate introduction. But I think of the species that I discussed, that's the one that where we have, I would say, some of the most questions about its replacement.
Thanks. And then just one really quick last question before we go. Nick Rasmussen asks, because he's not satisfied with all the ones that have invaded, is there a particular shrimp or crab species you're concerned with invading next?
No, I'm not. And that is because I'm holding out hope that ballast water exchange program is working, which has been in place since 2000. But human behavior is the wild card, of course. And I think people understand this in part because of some of the aquatic vegetation introductions that we've had that, of all things, aquaria releases are a very likely source for the next introduction, whether or not it's a fish or a plant or a crustacean here.
All right. Thank you so much, Kathy. That was a really interesting talk. Speaking of ballast water and aquarium release consequences, our next speaker is Andy Chang, who for the last 20-some years has been dealing with those in a number of communities, working at the Smithsonian Environmental Research Center. He's program leader there at their lab at Tiburon, and he's going to be talking to us today about various communities in San Francisco Bay with invasive species.
Thanks very much, Betsy. I'm going to go share my screen here. Thank you, everybody. It's been a really interesting morning so far. As Betsy said, we have been focusing on marine and estuarine invasion for a number of years here from our home base in Tiburon at the Estuary and Ocean Science Center. I'm going to talk today about some of the more recent findings we've seen in invasions in soft sediment and hard substrate communities in San Francisco Bay. And I want to first acknowledge a whole host of co-authors here. Gregory Ruiz is the head of our lab, and there's a number of folks who have contributed to the work here from our lab, and then our collaborators at Moss Landing, John Geller and his group. So, as we all know, there are a good number of non-native species in California. The number was pegged at more than 257. This is from one of Greg's papers about a decade ago. And here on the left side of the figure, we can see the number of first records of non-native species introduced to different regions along the North American Pacific coast, from Alaska, British Columbia, Washington, Oregon, California. And we see the number of species in black is the first record in California, and in white, first record not in California. As you can see, California has a good deal more than the other regions described here. And indeed, California has most of the newly detected species that have been found in Western North America since about the 1850s. So this is the number of newly detected species on the y-axis here across time since about the time of the gold rush. We could zoom in further. I won't do that here, but a large part of the species that show up in California first arrived in San Francisco Bay. And toward that end, the state, through CDFW's Marine Invasive Species Program, has stood up a program of surveys to assess the status and track the progress of non-native species entering California. And this is run through OSPR, the Oil Spill Prevention and Response Department. We survey a number of different habitats and fauna, hard substrate, soft sediment, and plankton, chief among them, with more limited surveys of the outer coast. And we've chosen these. We know that this is not completely comprehensive, but it will capture the majority of the species that are coming into California. Along the way, we are aiming to develop genetic libraries that will help us associate genetic sequences with species identification, which is a crucial step in making more efficient use of DNA for monitoring and tracking changes over time. We also have aimed to detect invasions over time and through varied environmental conditions, which I'll talk about a little bit further here today. So it basically boils down to who, what, where, when, and why, and how. among other things. So who are the invaders? What habitats have they invaded? Where have they invaded, geographically speaking? And when did they invade, and why are they so successful here? And what I'm going to talk about here is really focused more on the first three questions: who, what, and where, and to some extent, a little bit of when. Our general approach has been to use two sentinel bays, San Francisco Bay and LA Long Beach. These have a long invasion history, and they're well known as trade and commerce hubs for the state. There's a good number of other locations that have long invasion histories as well, but from what we have seen, the majority of invasions and invasion-related traffic and vectors tend to be coming in through these two sentinel hubs or bays that we've been studying. Their importance has varied over time, which is also a good way of tracking changes in vectors that may be bringing invaders in. We complement these two sentinel bays with a rotating survey of other bays and estuaries, and we go through a range of habitats, as I mentioned. So we've looked specifically at bays with commercial shipping and those without, as well as looking at stretches of the outer coast near these bays for a spillover of invaders from the bays to the outer coast habitat. And in addition to using traditional morphological taxonomy, as I'll describe here in a moment, we also use genetic approaches, including metagenetics and eDNA. All right, so let's get to it. With hard substrate, soft sediment, and plankton surveys at the core of what we do here, we use a couple of standard methods. In the upper right is a fouling panel. It's basically a very low-tech piece of plastic that provides a standardized surface for settlement of marine invertebrates. And we attach it to a brick for weight and suspend it from a rope from a dock or piling about 1 meter underwater and leave it for 3 months during the summer period. We've done this all over San Francisco Bay for 22 years, as Betsy mentioned, as well as a number of other locations in California. For soft sediment surveys, we have surveyed at several standard depths throughout the bay using a Van Veen grab, or I should more properly call it a young modified Van Veen grab, which has a few additional modifications to it. Deploy it from a boat and take a nice big bite of the sediment, bring it back to the lab and sort it. And our plankton surveys, which you see at the bottom left, I won't talk about in further detail, but I'm happy to talk about further if anybody wants to know. All right, so we are focusing here on hard substrate and soft sediment. Most marine invasions are on hard substrates, docks, rocks, pier pilings, seawalls, and rocky bottoms. These sites shown on the right are most of the sites that we have surveyed throughout the bay over the 20-some years. Of those, we have about 9 or 10 that we survey repeatedly year in and year out. So once we bring our samples back to the lab, whether it is from a fouling panel that has been sitting out in the bay for 3 months, or whether it is organisms that are sorted from a Van Veen grab for soft sediment, we have a standard workflow that we follow for identification. Bring it back to the lab, a team of people will go through and do a field level identification live under a microscope. And then we sort those samples and we get vouchers that are paired so that they can be used for both morphological taxonomy and for sequencing. And then we go through parallel workflows to arrive at a final identification through morphological taxonomy or through genetic analyses. With morphology, we have in-house taxonomic experts who will identify the sample, and then if necessary, they'll send it out and put out a call for help to do more specialized experts. On the genetic front, the vouchers will go through sequencing and then a complex matching procedure to arrive at a final genetically derived identification. And that end of things is handled by John Geller's lab at Moss Landing. I'm not going to talk about that much further here, except to have a brief digression right here, where I want to briefly say what we're attempting to do and moving beyond the limits of traditional morphological taxonomy. One of the main goals that we have, as I mentioned before, is to build a library that really helps us see what sequence equals which species. And we really need that in order to unlock the species identity of a sequence in a random sample. If we don't have that, then the sequence remains unidentified, one of the many thousands or millions of unidentified OTUs, or operational taxonomic units, in a given sample. So getting that library is key. And once we have that, we can use metagenetic approaches much more effectively and be more efficient in our monitoring. A key step in that is to determine how our current or traditional morphological approaches compare to these newer approaches. So that's something that we're working on. And we therefore view it as a complement to, but not a replacement for, the traditional morphological approach. Okay, so what I'm going to show here is a selection of results, recognizing that I can't be super comprehensive here. So let's start with a little bit of who and what from the hard substrate community. And I thought this would be a bit of an interesting addition following Kathy's talk. You're looking at paracarot and decapod crustaceans that are found specifically in association with the fouling communities that we have been studying. And this graph shows the number of species found at stations going from the Delta to the Golden Gate, starting with Sacramento and then moving all the way to the San Francisco Marina. So as you can see, the species richness per station generally increases as you move toward the ocean, which is the typical expected pattern in an estuary. And the number of introduced species also increases as we move toward the Golden Gate here. There are a number of species that are common throughout a fairly wide salinity range here. Species like the isopods Cnidotea labradorsalis. We tend to find them in many different stations throughout the estuary, particularly after a wetter year. There also are, I should point out, a number of species that are cryptogenic, meaning we can identify them, but we're not sure if they are native or not to this region. And there's a relatively tiny number of native species throughout the estuary. The unresolved category is typically reserved for specimens that are in poor shape or are juveniles, and we can't fully identify them. Okay, let me shift gears a little bit here to the soft sediment communities. This shows some of the diversity patterns in soft sediment communities over time, going from 2012 until about 2017 here. And these are the major groups that we've been finding in the soft sediment communities. And one thing I want to draw attention to here is the pattern of changes over time. We've got the number of species again on the y-axis, and then time within each panel from 2012 to 2017. I apologize for the differences in colors. I should have standardized those more. So cryptogenic is in blue, NIS, introduced species, is in light green here. You can see a large number of introduced taxa in the annelids and the arthropods, the two boxes on the left, as well as in the mollusks, all the way on the right. Not so much in the bryozoa or echinoderms, which are mostly brittle stars. There are 2 or 3 species. And then you can see that these change over time. And there's a couple of significant environmental events, which I'll mention in a minute, that we think are related to this. There's a huge drop-off in annelids in 2017, as well as in a couple other taxa. All right. Moving on here to where in low versus high salinity. I wanted to compare briefly here the pattern that we see in introduced species and hard substrate to soft sediment communities in the low versus the high salinity regions of the estuary. And the map on the right shows the high salinity sites marked with black dots. And then if you look closely, you can see the triangle showing our lowest salinity sites. There are more introduced species that we found in hard substrate, but not in soft sediment in the high salinity environment, which you can see on the left in the top panel. The bottom panel shows a greater number of introduced species in both habitats, which is an interesting difference between the two. I also wanted to touch on the ways that the introduced species prevalence can differ with conditions, to go back to the environmental differences across years for a moment. So this is a series of accumulation curves showing how we are getting more and more introduced species in our total pool of species as we collect more and more samples in a given year. The hard substrate communities on the top, soft sediment on the bottom, and these lines are colored by the previous winter's average daily outflow to the estuary in cubic meters per second. With the hard substrate community, and this covers about 18 years or so, and some of the lines are overlapping, you can see that we reach an asymptote generally fairly quickly, as is true for the soft sediment community, which means we are sampling the community pretty completely and getting a good idea of the invaders present. When you look at the colors of the lines, though, the blue line shows the highest outflow years, that's 2017 in both cases. We were finding fewer introduced species overall compared to the drier years. And it's an interesting reminder of why we need to sample repeatedly over time, because who knows, it might be a wet year or a hot year. Speaking of hot years, we had a marine heat wave a few years ago, otherwise known as the blob, which certainly sent a lot of warm water into the estuary. And there's a couple of interesting effects I wanted to touch on briefly here. First, we saw new invasions of southern species that we had previously only seen in San Diego and LA Long Beach, and those were coming up into San Francisco Bay. Time will tell if those persist, but it is an interesting start to what we could be seeing in the future. We also saw some shift in the dominant taxa in the communities and some interesting altered patterns of diversity that were rather different than what we had expected. I'm happy to talk about those more if anybody is interested. Lastly, I want to note that our species occurrence records are public. They are put up in the CalNEMO portal of our lab's Nemesis invasion system. So CalNemo, as you see here, the website, California Non-Native Estuary and Marine Organism Database. We aim to make this information public and publicly available, and our reports as well to CDFW also are available through the OSPO website. A couple take-home messages. So we found greater invader diversity and hard substrate across the salinity gradient. We did find that both hard substrate and soft sediment introduced species were reduced by that historic 2017 wet year. It's a reminder that they're all vulnerable to massive environmental events like that. And perhaps there's a management opportunity there when we've seen large natural reduction in the prevalence of invaders. Repeated standardized surveys like the one we undertake here really allow us to track these changes in invasion across a wide range of environmental conditions, and we hope will help predict the invasion consequences of management actions and responses to climate change. Looking ahead, we need to keep an eye on the climate ball, especially with regard to invasion and whether those southern species will continue to move up. And so we must ask ourselves if we're prepared for the changing risk landscape that these new events are posing. Lastly, it's good to take a look at how management actions are affecting invasion. So how are changes in ballast water management or vessel hull fouling, for example, impacting the arrival of new invaders? So these are all questions that our lab is working on as well. Thanks very much for listening, and thank you particularly to CDFW and the Department of Homeland Security for funding.
Thank you, Andy. Awesome to see that you guys are doing so much, especially with the pairing of DNA with morphology. I want to talk to you more about that after the session, but in terms of moving on to management actions, our final talk is going to be Jared Barr bringing it home with what we all want to do with invasive species, which is actually to manage them. And he's going to be talking about nutria today.
Hi, everyone. My name is Jared Barr. I'm a senior environmental scientist with California Department of Fish and Wildlife. Thank you all for coming to my talk. Today, I'll be discussing the progress of nutria eradication in California from 2017 to the present. This will be a good chance to share our successes with the program, with special attention to exploring annual trends. So kicking off with some intro material, nutria are a large semi-aquatic rodent. Adults range in body size between 1.5 to 2 feet, and they weigh between 15 to 25 pounds. They inhabit fresh and brackish waters, and they're native to southern South America, and they were introduced to the US for the fur trade in the early 1900s. From there, feral populations established from escaped nutria. They were, however, declared eradicated in the 1970s from the state, but then in 2017 they were rediscovered on a managed wetland in Merced County. So nutria are a high priority invasive species because of the extensive damage they do to both habitat and infrastructure through vegetation loss and burrowing. They consume up to about 25% of their weight each day in vegetation, and they're wasteful feeders, so they destroy up to 10 times the amount actually consumed. This leads to severe erosion, loss of plant cover, loss of organic matter for soils, and eventually conversion to open water. This exacerbates invasive plant infestations, and burrowing damages infrastructure and levee systems, causing severe erosion, sedimentation, and impacting streams and rivers. So our eradication strategy in California has been adapted from the 5-phase strategy that was developed by the Chesapeake Bay Nutria Eradication Project. That project successfully eradicated nutria populations from over 250,000 acres of habitat on the Delmarva Peninsula in Maryland. So first, we pursue land access throughout the entire region, which currently encompasses 11 counties. Then the region is processed using a 40-acre grid overlay to help track our progress. So each grid cell is tracked with 5 phases depending on nutria presence. These phases include survey, knockdown, mop-up, verification, and surveillance. So for the survey phase, our primary methods for early detection include grid cell assessments and deploying and monitoring game cameras and platforms. These cell assessments consist of comprehensive sign searching and classifying of habitat. For the knockdown phase, this is an intensive trapping effort if nutria are detected in the cell during the survey phase. And then for the mop-up phase, this is when nutria densities in the cell are at 0 or near 0, and we need to go in and kind of clean up the remnant individuals left in that cell. For the verification phase, this is verifying that densities are at 0 for nutria in that cell, which is more surveying effort. And then for the final 5th phase is surveillance to kind of ensure that eradication in that cell has been successful. To get some more context for the past 5 years, let's take a look at the history of nutria eradication efforts in California. So this is a brief timeline of the past 5 years. In 2017, the first nutria in California was discovered, and local biologists responded rapidly and began surveying. They quickly realized that the infestation was larger than anticipated, and in 2018, an incident command system was initiated. The ICS was an emergency response consisting of redirected biologists. Partnerships and collaborations began during this time as well, with agencies such as CDFA, USDA, U.S. Fish and Wildlife Service, and DWR, and others. In 2019, we received grants from the Wildlife Conservation Board, U.S. Fish and Wildlife Service, and 2 from the Delta Conservancy. These allowed us to expand our operations into a fully dedicated field crew of about 40 staff. Also in November of 2019, the Nutria Eradication Program within CDFW was established. So 2020 was the first full year of operating with an established program. So that year is an important benchmark to keep in mind when examining trends, because our survey effort and coverage greatly increased during this period. 2021 is also a critical snapshot in time, given the ability to directly compare our efforts year to year with 2020. Let's take a closer look at the current state of eradication efforts. So we've done assessments on over 1.5 million acres. We have entry permits with over 4,000 landowners, which is just over 8,600 parcels in 11 counties. We've deployed over 5,200 camera stations, of which 900 are currently active. And we've detected nutria in over 500 unique grid cells and deployed over 6,500 trap sets for a total of over 84,000 trap nights. And as of March 8th, we've taken 2,862 nutria from the population. And here's the current known geographic extent of the infestation. As I mentioned, a total of 2,862 animals have been removed, but by county, we've taken 1,856 from Merced, 876 from Stanislaus, 110 from San Joaquin, 12 from Mariposa, 7 from Fresno, 1 from Madera County, and we have had a single confirmed detection out of Tuolumne County from a photo submitted to us by the public. But following surveys and extensive monitoring in the area, we haven't been able to relocate the animal since. Heat maps are a great way to visualize the spread of invasive species and the effort involved. The following maps illustrate the least and most nutria-dense grid cells in regard to take of nutria. The scale on the left of sparse to dense is actually a range of cell densities from 1 to 156, since our densest cell for a single year contained 156 nutria. We wanted to keep the scale consistent across years, which is why we used that densest cell in a calendar year. These maps will highlight the geographic depletion and/or accumulation of nutria across years. So as you can see for 2017, nutria were taken near San Luis National Wildlife Refuge and also on the Merced River. Here's the map for 2018. We saw more take on the San Joaquin River corridor and Merced River corridor. Also, this is the first time we actually detect and take animals in the Delta, specifically in that southern part of San Joaquin County. And here's the map for 2019. And here's the heat map for 2020, which was the first year of the established program and a subsequent increase in resources, manpower, and survey effort. And here's the map for 2021. Note that the heat maps show that take has occurred roughly in the same areas throughout the years, even though we have monitoring equipment and ongoing surveys throughout a much larger geographic area encompassing 11 counties. When compared to 2020, which was the first year of the program and an increase in resources and manpower, we definitely see some depletion of nutria within the main areas of infestation. These large metapopulations are in Merced and Stanislaus counties, and you can see where we're starting to deplete those sites. To get a more detailed picture of where nutria have been known to occur in California, let's look at a set of detection maps. So the following set of maps focus on take plus detections. The points on the map symbolize the density of animals within each 40-acre grid cell, as you can see from the legend on the left-hand side. This is our map of detections and take for 2017. In 2018, staff discovered and captured more nutria on the San Joaquin and Merced River corridors. Note the blue dots are detections only, with no take occurring in that cell. And here's 2019. Note the single detection up near Stockton. This animal was never captured on camera again, and it was thought to be a dispersing adult male. In 2020, we were able to increase survey effort and coverage, so you'll notice more low-density cells on the map. There are also some high-density cells near San Luis Refuge, along the San Joaquin River in Stanislaus County, and also along the Merced River. And in 2021, we have fewer hotspots, more low-density cells, and more detection-only grid cells. This indicates that nutria are being knocked down with some effect. An increase in detection-only cells and low-density cells is a positive sign. As we deplete the nutria population, and especially these large family groups, we are seeing an increase in dispersal by remnant individuals that are in search of conspecifics and other nutria family groups. Often we'll have one animal dispersing through 6 or 7 cells until we actually are able to remove it, which is why you see an influx of detections but no take for 2021. Also, it's important to note that 3 nutria were taken at the beginning of 2021 in the Delta, or San Joaquin County, but none have been taken since, even though surveys and monitoring is ongoing throughout the Delta. There's no evidence to suggest a breeding population in San Joaquin County, just occasional dispersing animals looking for conspecifics. Also, historically, 364 cells have entered the knockdown trapping phase. Of those cells where knockdown trapping has occurred, 263 of them, about 72%, have been subsequently transitioned to the mop-up phase. The remaining 101 cells in the knockdown phase are still being trapped or are currently being evaluated for transition to mop-up. Another 67 cells were transitioned directly from the survey phase to the mop-up phase due to their proximity to cells with nutria taken or the nature of detections and/or take within the cell. Evaluations are ongoing for transitioning these areas to the verification phase. These maps don't tell the full story, so let's dive more into the numbers. This graphic illustrates annual nutria take and survey coverage among years. The gray bars represent the number of distinct cells surveyed. So we have drastically increased our survey coverage over the years, with 2021 having over 5,000 distinct cells surveyed, which is equal to about 203,000 acres. The red bars illustrate the number of nutria taken. Notice that our nutria take nearly tripled for 2020 when compared to 2019, which makes sense when you take into account our increased effort and boots on the ground for that year. Then, take dropped nearly by half between 2020 and 2021, even though our survey coverage and effort increased. We saw a 48% decrease in animals taken and a 10% decrease in number of unique grid cells occupied. This indicates that our efforts in the last couple of years have yielded positive results. We'd like to continue to see sustained or growing survey effort and nutria take continue to decrease. So we believe 2020 was our population peak, and we're currently on the downslope. For the beginning of 2022, we've taken just 63 nutria, which is roughly half the number taken at this time last year. Here's a closer look at nutria take throughout the years. The middle column is the number of cells with the nutria taken, and the right column is the average number of nutria taken per trapped cell. For 2021, both the geographic extent and the density within cells shrunk. Again, we'd like to see this trend continue, and we have some strong indicators to believe that 2020 was our peak in population size and that we're currently on the downslope. So this figure shows the age and sex breakdown of captured nutria throughout the years. Blue bars are male and orange are female. In this graphic, each year is treated as a separate snapshot in time where percentages add up to 100 for each year. So we ascribed 4 different age classes to captured nutria. 1 is less than 2 months in age, 2 is 2 to 6 months or juvenile, 3 is 6 to 14 months or subadult, and 4 is greater than 14 months or adult. As you can see, there isn't much of a trend here among years, but within years you can see a pattern of juveniles making up most of the population, followed by subadults, adults, and young. As of this month, we've removed over 3,100 fetal nutria, and in California, the average litter size is about 5.8, and roughly 40% of all captured female nutria are pregnant. Even with the positive results we've seen these past few years, we still have some obstacles to overcome. The most important challenge is gaining access to some high priority sites where we assume nutria occur, but to date we've been precluded from entry. A lot of these are private lands, and unfortunately without adequate access, we simply don't know what's on the ground and if they're harboring nutria. Aquatic invasive plants have also been precluding us from entering areas that have or had nutria present. Water hyacinth has exploded in the last year or so, likely due to drought conditions, and in some cases we can't even get back to our trail cameras that were placed not even a month prior. Unfortunately, this has been a problem in the 3 counties where our field work is most important, but we're currently working closely with the Parks Department, Division of Boating and Waterways, so we can continue to effectively do our jobs. As a reminder, nutria are wasteful feeders, so they exacerbate invasive plant populations like water primrose, water hyacinth, and parrot feather. So I'd like to thank Valerie Cook and Ruth Spell for helping co-author this presentation. And we'd like to thank all of our partners and collaborators throughout these past few years. We have so many people to thank, many of which I wasn't able to include on this slide. And we'd also like to thank the IEP organizers for allowing us to speak at this workshop. And with that, I can take any of your questions.
Thank you so much, Jared, and thank you for making a nice circle back to the beginning of the session, back to the aquatic veg, and looking at some of how the invaders are interacting with each other in ways that we didn't anticipate. Anyone have any questions for Jared right now? Come on, guys, you've got a live nutria biologist on the line. Jeffrey Holt, do you want to ask yours?
Yeah, thank you. You actually sort of answered it, but I know a lot of those areas down in Merced County are duck clubs adjacent to some of the federal and state lands, and it doesn't sound like you're having a lot of success to get access to those, or you kind of speak to some of those challenges?
Yeah, so we do have quite a bit of access with duck clubs down in Merced County. We've been working with those folks for a number of years, since at least the beginning, 2017, 2018, because that was the kind of epicenter of the infestation and the beginning of, the reintroduction of nutria in California. So we work with those folks extensively. We have quite a bit of access down in Merced County with duck clubs and gun clubs. And yeah, we do kind of switch off seasonally between the grasslands area where there are quite a bit of duck clubs and with the Merced River. So typically when water is drawn down, we will go back to the grasslands because it's easier to find and capture nutria when the water is drawn down, and B, it's no longer duck hunting season. So typically when we do have access to these duck clubs, it's of a seasonal nature.
Great. Well, I think we're just about to our break time. So I just want to thank all of the panelists for coming and sharing their research with us. I've really liked how we've gone sort of the whole cycle of invasive species research, from detection, identification, characterization of populations, studying some of the factors behind them, all the way through to some of the management efforts. And thank you all for speaking. Thank you all for attending. This has been a really fun session. And if anyone has questions, please feel free to follow up with the speakers, and please tweet about this.